Good evening, Professor. This is my personal timetable. In the next week, I have planned four different kind of tasks. The first one is urine biosensor research. This already started the draft paper, and I have also explained you my uh, research roadmap of the urine biosensor paper. And also, I have to start again experiment for the capacitance measurement, and I'm also simultaneously looking for the zeta potential measurement that I already talked with the Ryonsang to search this instrument. Is there any university? So we found that the Yonsei University have the zeta potential instrument. So in this week I have to do the official booking, and then meanwhile planning for the going to the zeta potential measurement. The second one is corona biosensor research. Me and Prashant has already started, and in this week we have to show you the results. And the third one is AAO biosensor research. I have discussed with Bruce and also did the FTIR measurement in the last week, and that I have already shown in the next slide, and also start the draft paper, and as well as also simultaneously working with the AAO nanopore structure to deal with the graphene oxide and the biological elements. And the fourth one is the meeting with the Jacob and Bruce. So Jacob is already started working with the IPD with the Prashant, as well as Bruce will do the work focusing on the draft paper as well as the research. So last week we did the FTIR characterization of the glass chip that we have that we act as a bear. So we focus to check the first the what is the basic chemical bond between the Ag protein and the graphene oxide and the specific antibody and antigen. So we present here six different types of FTIR results for the selectivity measurement of the biocomposite, that is the bare one, and the second is Ag protein that you have given us before, and the graphene oxide is already presented in the lab. It's a single layer graphene structure is we bought, and it's an aqueous solution to easily deposit it on the surface, and we use the layer by layer method. So it means layer by layer means the first we put the graphene oxide, and then we are waiting for drying, and then we put Ag protein. This is the LBL method. It's a layer by layer. And the fifth one is mixing method. It means we mix together graphene oxide and the Ag protein mixed together, and then we immobilize or do drop cast on the surface in a specific amount, like we use normally 2 microliter, and as per the requirement of the surface. And also we use here one different kind of antibody, just checking how it perform with the antibody and antigen. So we use here antibodies 1H12, that is related with the urine biosensor antibody, and the other is antigen is related with the specific antibody. So here we are. So this is the FTIR graph of the three different kind of uh, statistics. So we first is the bear. So as you can see, the bear is a black color line, so it comes like this and like this, the black color one. And the Ag protein is a red color, so it's the transmittance is higher, but the wave number, in terms of wave number, is not specifically to check the at what segment, at what wave number we have to get the chemical bond. But in this case, the graphene oxide, it equally parallel to the bare one. So let's talk about the other, like what we are the next did. So we compare with the bare. So the second one is the graphene oxide First, we put the graphene oxide, waiting for drying, and then AG protein, and then wait for drying. So it means layer by layer method. So, and the third one is mixing method that I already explain you about the what is the difference between LBL method and a mixing. So mixing, it means we did mixing together of graphene oxide and AG protein, and then we put 2 microliter of the mixture on the surface. So this is the graph. So it means there is no as such specifically combination, but when we zoom in that particular segment, we found that the red means mixing is more getting transmittance as compared to the layer by layer. So we focus on the what is the next if we put antibody and antigen. So so this is the graph that we define that the mixing is good or the LBL method is good. Either we treat it with the EDC NHS antibody. Is antibody is a 1H12 antibody, and antigen is the specific antigen that is correlated with the antibody. So here I focus the segment. Then we found that the EDC, if we use EDC, then we got the transmittance like the Bayer. This one is LBL method of that particular composite, and then we put EDC and antigen, then we found this peak. So if we compare with the mixing method, why I am comparing, because to check the which one is more suitable, either LBL or mixing. So then so I have shown here the mixing one also. So we mixed together and then we treated with the EDC NHS. So we found that the transmittance and the graph is quite different. So there is no as such mixing results is quite good. So then we focus on the LBL method and we go for with the LBL method. So also here we compare the results without EDC. In the last slide we focus with the EDC, where we use EDC NHS. It's a cross linker. So for EDC NHS, and then we put without EDC. Then what is the difference? So the difference is quite not good results, because here we use EDC, then we got the like the the in one segment we got the same specific peak, but in here we didn't get any good results. So we found that without EDC is not much suitable. Sir, and this one is the NDAC project. It's a AAO nanopore project. So this is my main research concept, that what I make it. So we use here first our the the substrate, and then we use the chemical with the cross linker, and then we use here EDC NHS to cross link between this the graphene, and then we put the antibody and to deal with the antigen to check the disease marker in presence of BSA, is bovine serum albumin. So it's to clean up the unwanted antibody from the surface. And so this is the plan. And next we found that the SiO2 surface cleaning process with the Purina process, it's like means if we didn't make the Purana solution, then our surface is hydrophobic. So we found that if we did the Piranha solution treatment, then our surface is act like a hydrophilic surface. So this is why we use this step, to make the surface hydrophobic to hydrophilic. So we have to we did the this chemical composition and do treat it with the 200 Celsius for the 20 degrees 20 minutes to make our chip hydrophilic. So and this is the method. This is the another LSPR graph to show that it's result comparison of graphene oxide with AG protein. So it's AG protein with the AG protein, so we didn't get any. Um, it's basically the graphene oxide, and then it's the AG protein. So the AG protein result is quite good rather than the graphene oxide, and we check the literature. So there is no as such peak of the graphene oxide. So we go with the AG protein. So AG protein is the graph is good because it comes from the 400 to 450 nanometer, and then we mix together. It means like the graphene oxide and the AG protein mix together, and the AG protein single and to graphene oxide to see the result here. So the graphene oxide peak is not any specific wavelength and then we graphene oxide with AG protein, so we got good peak at a specific wavelength, and then the AG protein is also good as compared to the graphene oxide. So here we just compare all the results, like what is the difference between if we use the mixing method or the LBL method. So the AG plus protein, AG plus protein is the blue line, so it goes like this, and the graphene oxide is the dotted line. So as I already mentioned before, and the graphene oxide with AG plus protein is this the another gray line is like this, so it correct 5, and then the mixture is more higher. So we found that the mixing of the chemical is more efficient results rather than the layer by layer. So we go with the mixing, as well as also we have seen that the mixing idea of in a case of FTIR is also good. So sir, our next step plan to using AA platform with graphene to check LSPR measurement, because we did with the SiO2 glass, so we also perform with the AAO platform, and then we conjugate with the biological elements like antibody and antigen, and then with the also use the different kind of polymer. So I have to do the literature survey first, and then we put use the characterization like AFM, SEM, TEM, and the zeta potential measurement also to check the biocharacterization of the samples. So this is the zeta potential measurement. Is I have talked with the Ryun Sang to search this instrument in any institute or in university. So we found that Yonsei University having this instrument. So I have to just book this instrument, and in the next week I have to go with the samples to measure the particle size to conductivity, ionic strength, sample concentration with our samples to check is there any changes or is there any particle size differ from or in each case of a step, and then I have to present my results in the next lab meeting. So this is the urine PSA biosensor that we already followed this step to check the AFM, SEM and the capacitance value. In the next week I have to present my results of the capacitance measurement. And so this is my research roadmap that I already done the uh, capacitance measurement, but it's not good, so I have to repeat it again, and simultaneously also working with the title, abstract and schematic figure. So Ericsson is doing the schematic figure, and also the Jaya also did some figures, so I have to just guide them to make a appropriate kind of schematic figure. So in the sir, I have also written the some introduction part as well, and in considering of result and discussion, so I have to work little bit more in this section, and the data optimization and the funnel setup I have to check the according to the result and discussion part. Sir, review article I have already submitted to you and waiting for the comments. And according to your comments, I have to do the correction and then I have to submit to you. So this is the coronavirus biosensor research. It's already on the way, and hopefully I will present you the results as well. So this is the research AO plan that I already planned with the Bruce to do the to using the AO platform to check with the measurement. Sir, and this is the paper writing plan of the urine PSA biosensor. Thank you, sir.